So as we know from previous weeks, there are many differences between prokaryotes and eukaryotes. And talking about their differences in transcription is really no different. So to begin with, in this section what we're going to do is we're going to focus on how eukaryotes perform transcription. So to begin with, eukaryotic transcription is going to require that chromatin to become uncoiled. And the reason for this is it's going to make the DNA accessible to the RNA polymerase and other regulatory proteins it might need. This transition is referred to as chromatin remodeling. So if this DNA is tightly wound up, and it's going to be inaccessible physically for that RNA polymerase as well as these other transcription factors. It just needs to be more loose and accessible for these proteins to come in and interact with the DNA. However, eukaryotic RNA polymerases are going to also rely on transcription factors, or TFs. What they will do is they will scan and they will bind to DNA, and they're very important for turning genes on and turning genes off. In addition to this promoter region binding the transcription factors and RNA polymerase, there are some distant regions, they can be distant or nearby, called enhancers and silencers. And as their names suggest, they can either enhance gene expression or silence gene expression. And so they can both control this transcription. So some more differences. Eukaryotes are going to possess 3 types of RNA polymerase. Each of these is going to transcribe different types of genes. The most abundant is probably going to be RNA polymerase II. RNA polymerase II is responsible for making the mRNA as well as snRNAs. RNA polymerase I is going to make the rRNA, which is responsible for the ribosomal RNA. RNA polymerase III is responsible for the 5S RNA as well as the tRNAs. So as you can see, again, this RNA polymerase II, that's the one we're going to find that's going to be transcribing most typical genes. The RNA polymerase II promoters have a very core promoter element, and the promoter is going to determine where that RNA polymerase will bind to the DNA and where it physically begins copying that DNA into RNA. It is also going to have similar elements within the sequence of the DNA, such as the TATA box. The TATA box is going to bind proteins, this one specifically a TATA binding protein, or TBP, of a transcription factor, T2F2D. And this will determine the start site of transcription. Several other of these regulatory DNA sequences, you could call them proximal promoter elements, which would be nearby to that promoter. Enhancers and silencers, they can either be nearby or further away. They can even actually be within a gene itself. All of these will influence the efficiency or rate of transcription initiation. Some of them can act very far away. They can be quite a ways downstream, and they can modulate this transcription from like I said, from quite a ways away. So identifying these is really very difficult for scientists, because how do you know if something, 100 kb downstream is going to affect that particular gene? What they're doing overall, though, is to increase or decrease transcription. They're going to allow a cell to respond to things. Okay? Maybe a cell doesn't need a particular gene product. It can turn it off. Maybe it suddenly will need something. It can start to turn it on. And this way allows the entire organism to respond to various different things. So talking a little bit more about these transcription factors, there's basically 2 types or categories of transcription factors. All of them, though, are going to help promote RNA polymerase binding and initiation of transcription. If something is called a general transcription factor, it is going to be absolutely required for all RNA polymerase II-mediated transcription. The other type are going to be transcription activators and repressors. And they are not absolutely required. However, they can definitely influence the efficiency or the rate of this RNA polymerase transcription initiation. So, once all of the components assemble around the promoter region, RNA polymerase will open up and separate or denature the 2 strands of DNA. And the reason for this is so that the template strand can pass through its active site. And this is going to allow RNA synthesis to proceed. As this transcription proceeds, the enzyme will scan and move along the DNA until it reaches the termination sequence. The enzyme, the RNA complex, everything will then disassemble and separate. So there's some more differences between the prokaryotes and the eukaryotes in transcription is that in a prokaryote there is no nucleus. So any mRNAs that are made can be immediately translated. And we're going to talk more about that in chapter 14. Eukaryotes, though, because there is a nucleus, it's a lot more complicated. They also require processing, several different processing events to happen in order to produce the mature mRNAs. Only then are those mRNAs allowed to leave the nucleus and allowed to undergo translation. These processing events include addition of a 5' cap, addition of a 3' tail, and excision or removal of introns.